is he's also my mentor, so he can interrupt me and like fill in on anything I missed. All right, so this summer I worked on a project called AAV9 for treatment of error of cholesterol synthesis. So before I tell you what AAV9 is or what an inborn error of cholesterol synthesis is, I think a good introduction would be a cartoon on genetic inheritance. And the main takeaway from these cartoons is that we inherit traits from our ancestors. Some can be good. If both our parents are super geniuses, our IQ will most likely be above average. On the other hand, if diseases such as diabetes, atherosclerosis, heart disease, Alzheimer's, Huntington's, they can all be passed down from parent to offspring. And that will be the intro to the talk today, which is on Smith-Lemli-Opitz syndrome, which is also a genetically inherited disease. So my project the resolution is weird for this. I can manage with it. Okay, so minor technical error. So my project focuses on the brain. So let's take a zoom in. So in the brain there are neurons, so I'll probably talk about it already, so I'm not going to go deep into it. And in neurons, in the nucleus, there is DNA. So DNA codes for proteins, and proteins perform all the metabolic functions in our cell. Think of it as an instruction manual that tells your cell what to do, what to make, so that you can be you. And with SLOS is basically this DNA goes through mutations. This DNA goes through mutations so that it can no longer produce cholesterol. And people who have the mutations, they can pass it down to their children, and their children can also pass it down. So how common is it? Well, it's actually pretty common, surprisingly. It's recessive, so like blue eyes and no ridge on the hair right here. It occurs maybe 1 in every 30 people in Caucasian populations. And that translates to about, perhaps 1 in 10,000 births to 1 in 60,000 births. So pretty high. That's a takeaway message. So exactly what happens? So remember when I told you earlier that DNA makes proteins? Well, the exact protein that is made is called 7-dehydrocholesterol reductase. It's called DHCR7 up there. And what it does is it converts 7-dehydrocholesterol to cholesterol. So what happens if you don't have it? You don't make the protein, and you don't make cholesterol. And the one difference between cholesterol and 7-dehydrocholesterol is that double bond there, for those who don't understand, it's just one line. It's one line, one double bond, and yet it makes the world's difference. It can be the difference between you living a full life and you dying before the age of 18. And because 7-DHC, it can be toxic if accumulated in very high amounts. Okay, so what cholesterol does is, it's very important. It's in bile, it's an emulsifier, so it helps water, fats mix together, aids in digestion. Cell membrane, cholesterol is in the cell membrane, it helps give it its structure. Hormones, if any of you remember cortisol, aldosterone, testosterone, estrogen, you couldn't live without those hormones, and yet they're all derived from cholesterol. Hedgehog signaling. Imagine you were the little embryo when you were first made. Hedgehog signaling is basically the processes, perhaps regulated by cholesterol, that tell you each part of that little embryo whether it's going to become an arm, a heart, a kidney. So without cholesterol, you get, okay, so it says SLOS clinical manifestations vary widely in type and severity. That's what it's supposed to say, but it's cut off. So I'm just going to read it off since it's hard to see, but it says fused toes. So with SLOS, the most common is the second and third toe are fused. And another one is hypotonia. You can see with that baby, very weak muscle tone, cannot raise its head, cannot move. When I was young, I couldn't move anyways, but that's a different story. Now, and with that little boy down there, he looks normal, but if you look at him at eye level, you can see up his nostrils. That's not supposed to happen to babies. And his ears are lower along the side of his face. And the cleft palate leads to cleft lip very often, and polydactyly. The sixth finger that comes with SLOS often happens towards your pinky. It's called post-axial polydactyly. And of course, behavioral and intellectual disabilities, I don't have any pictures for that, but it's there, similar to autism. Now, SLOS has very few treatment options, which is why I plan to explore this area. Cholesterol supplements, fancy word for more eggs and seafood. Statins prevents you from building up that toxic 7-dehydrocholesterol, very important. Surgery doesn't actually cure it, but it certainly helps with the congenital malformations. And you'll notice that none of these are actually cures. However, gene therapy, that really had to be in. So, gene therapy, the gist of it is you take a gene. So in this case, the one that will code for 7-dehydrocholesterol reductase, the normal gene, and you package it into a virus. And then the virus, you'll inject the virus into the patient who cannot make his own cholesterol or cannot make enough of it, and then that virus will give the normal functional gene to the patient. And in this case, because our patients are mice. And our mouse models are FBB models. And where you use them is one, they have huge litters. And for the most part, they're fairly healthy. The other thing is they reproduce a lot. So they breed a lot. And what happens is we breed them together, and each of them has varying degrees of severity in SLOS. And we treat the subset called delta T93M mutants. And you don't have to remember it, but it basically means it's the closest representation, the most severest form of SLOS in our mouse models. And we inject them with a virus called AAV, adeno-associated virus. And what we do is we transfect human embryonic kidney cells with the dehydrocholesterol reductase plasmid with AAV DNA, and we should get viral DNA that contains the DHCR7 gene packaged into the virus. And this virus we can inject into our mice. Now the problem is we're trying to treat the brain, and you can't just feed someone the virus. You can't just inject into the bloodstream, mainly because of the blood-brain barrier. Blood-brain barrier prevents most substances from crossing, and such as cholesterol, for example. You can't just eat cholesterol and expect it to go to the brain. It's not going to happen because cholesterol is too big, even though the blood-brain barrier allows most fat-soluble substances to cross. AAVs, the ones we have been using called AAV8, cannot cross the blood-brain barrier. So the virus, if we inject it into the bloodstream, can't reach the brain because it doesn't cross that barrier. But water, ethanol, glucose can cross. Other words for it are water, alcohol, and sugar. They can cross. So how do we bypass it if we're using AAV8? A good technique is known as intrathecal injections, where you inject into the spinal cord. And because the spinal cord is continuous with the brain, the virus can reach the brain. Or the nerves in the spinal cord can lead into the brain. And that's how we can treat neurons and glial cells. So what we do here is, in the past, is we take 2-3-day-old mouse pups and we inject them at the thoracic 1 to 4 spinal columns. And the results have been promising. Is based on this ratio, I'll describe it first, is the lower the ratio, that means the more cholesterol that's being produced. So the lower the bar, the better. So according to the graph on the left, because the bar goes down, it seems that perhaps intrathecal injections lead to promising results. But this is not normal. This is not normal cholesterol levels at all. Because in normal mice, you wouldn't see that bar. That bar would be almost invisible. So here's where AAV9 comes in. Why would we do AAV9 if AAV8 seems to work? And part of the reason is AAV9 may cross the blood-brain barrier, or it does cross the blood-brain barrier, apparently. So the hope is that when we do inject it, AAV9 should cross the blood-brain barrier to deliver DHCR7 genes to all the neurons so that they can produce their cholesterol at the most important stage of mouse's lives, which is at the very beginning, when cholesterol is needed most when it's entering the nervous system. And once we inject, we'll compare the results to the intrathecally injected AAV8, and we'll see which one's better. So that's the timeline for the next 3 months. I'm still making virus at this point, clearly. And acknowledgements, I'd like to thank my mentor, Dr. Gordon Watson. He's also the moderator. My postdoc, Dorothy, another member of my lab. The SURF, people who run the SURF and make it possible. I really learned a lot from this experience, and I would come back any summer to, to work on this. And my UC Berkeley mentor, Helene Ratsiraswan, who made this possible for me as well. Thank you.
Are there any questions for Sean?
Have you thought about using some sort of neurotropic virus like a herpes or Epstein-Barr to package your DNA and see if it's able to be effective at infecting neurons?
Well, the thing is, when we're picking a virus, we have to be careful about integration. And integration is it inserts the gene into the actual host genome. And we don't want that because that can cause cancer. AAV8 is a very good option because AAV does not have an integrase. So it does not insert the gene into the host genome. It actually stays outside the host genome as an episome. And that's how the gene is transcribed. So it avoids the risk of actually causing cancer. And there have been many cases where adenoviruses have actually caused cancer and end up killing the patient.
Any other questions?
Does this disease totally knock out cholesterol production throughout the body, or is it differentially?
That's a good question because it varies. You can have the same mutations, and yet you can have varying degrees of cholesterol in your body. So the pathogenesis of the actual disease is not very well understood.
You can't get a total knockout because
Yeah.
not having cholesterol is incompatible with eukaryotic life. Are there any other questions? Could you comment on whether or not your AAV virus is still an infected virus or not?
Infected? No.
Infected.
Infected? No, it's not. Mainly because the replication and capsid proteins are no longer part of the virus anymore. So it cannot, once it delivers a virus, it's not going to infect any other cells. So it just delivers and it's done.
Are there any other questions? Thank you.
Okay.